All right, so now we got some good stuff going on today. We're doing a nucleophilic acyl substitution. This one's going to be a little bit more comprehensive because we actually do know a couple nucleophilic acyl substitution reactions, but just a general mechanism for you right now. If we have a carbonyl compound with a Z group on it as well as an R group, remember that Z group is a leaving group, something that's a little bit more electronegative than carbon or hydrogen, and we'll have a nucleophile substitute that, and we'll get our new carbonyl compound. So we've seen this with acid chlorides and diol creating aldehydes. We clearly substituted a chlorine with a hydrogen. That's an acyl substitution. So the acyl group is just the RC double bond O portion. Um, just to let you know, and Z must be a good leaving group. Or just a leaving group in general. Sometimes it's not very good, but we do things to make it better. So nucleophilic attacks, and we create that negatively charged oxygen, which comes back down to create that pi bond and kick out Z, the leaving group. So another example that we saw was with an organocuprate. Remember diol and the organocuprates, they're not as strong as lithium aluminum hydride or the other organometallic reagents like Grignards or organolithiums. And so you end up with ketones and aldehydes with those guys. Okay. So when we're looking at what's going on with this particular reaction, remember Z as a leaving group is going to be viewed as Z minus. We also have this negatively charged nucleophile. We have the negatively charged oxygen in there. And so we want to look at the basicity or the acidity of the conjugate acid. And remember that the weaker base means it's going to be a better leaving group. So we can break this down into 5 different classes of compounds. So we've got our acid chloride, we've got our carboxylic acid, we have our amide, we have anhydride, and we also have esters. Now that's in no particular order, but I would like to put them in a logical order for you. One thing we need to note is that all of these guys, they have leaving groups. So they're pretty much just not hydrogen or carbon containing groups. Another set of compounds, which is interesting, they're pretty much cyclic esters. They are called lactones. And the one on the left is called a gamma lactone, and the one on the right is a delta lactone. The way this works is because we can count alpha, beta, gamma, or alpha, beta, gamma, delta from the carbonyl group, and then cycle all the way around to see where it's connected to the oxygen. We also have cyclic amides, and those would be lactams. So I like to remember these as lactams, like the AM is amine. And the naming system is pretty much the same. We have a beta lactam and a alpha beta gamma lactam because that gamma carbon is bound to the nitrogen, which completes the cycle. So when it comes to structure and bonding for these guys, just a quick little reminder, the structural part of basically the RC double bond O is the acyl group, so that's why we're doing acyl substitution, because we're substituting the Z for something else. So we've got acid chlorides, anhydrides, carboxylic acids, esters, and amides. Now this is in order, and this order here is in order of excuse me, leaving group ability. So as we go from left to right, we have a chloride that would leave. or we have an acetate that would leave, or a hydroxide, or an alkoxide, or an amide that would leave. So as we go from left to right, we have a decreasing reactivity because those circled groups don't want to leave, and that inherently means that they are increasing in stability as well. So this stability is going to be due to an increased resonance stabilization as well, and as well as in addition to that decreased leaving group ability. So here we go. What I mean about the resonance. So if we have the lone pair on that chlorine that can create that resonance structure here, and then on the other extreme we have an amide. The nitrogen has a lone pair that can also do this resonance structure, creating both a positive on the chlorine as well as that nitrogen. But when it comes to resonance structures and the general population of each of those resonance structures on the right hand side, the amide, that's actually a major contributor because nitrogen is a good pi donor and it's less electronegative than chlorine. The one above that is actually not a very good resonance structure because we put a positive charge on a very electronegative atom. So what I want to do next is classify each of these leaving groups according to their pKa's, written in blue. So remember, their pKa's are directly related to their leaving group ability. So since we have a chloride leaving, the chloride's Cl minus, but the conjugate acid of that is HCl. HCl is a very, very strong acid and has a pKa of -7. Since it's such a strong acid, that means that chloride ions are very weak bases. Since they're very weak bases, that means they're excellent leaving groups. And then we can further classify the rest as +4 for the acetate, which would be acetic acid, 15.7 for the OH, which would be water, about 17 or 16 for the alcohols that are leaving on the esters, and then about 39 for the amines that are leaving on the amides. Okay, and I've drawn out a scheme more or less of the acid chloride and how the leaving group and its relationship to HCl below. Now, what I want to do next is use this little chart to classify our IR stretches. So what we're classifying is the carbonyl stretches. And as we go from left to right, we'll notice that the values actually decrease. And so this is actually related to the resonance structures. And so I'll go over that in just a second, the details on that. It's pretty straightforward, but it's related to physical properties, nomenclature, as well as spectroscopy, right? So I want to let you know you got to handle the nomenclature and physical properties on your own, and then we can do spectroscopy together, even though it is pretty straightforward as well. There's some minimal explanations for you. So back to the IR and the CO stretch, they range from 1600 to 1800, right? 1600 was for the amides, 1800 was for the acid chlorides. Well, this varies due to resonance and their contributions to resonance structures as those Z groups. So back to this acid chloride resonance structure and the amide resonance structure. If we have a higher population of the amide resonance structure on the left versus the acid chloride resonance structure on the left, then we would have a higher population of a compound or a structure that has more single bond character. And so this, remember, is related to having lower stretching frequencies on an IR. So that's why we have 1630 to 1680 for amides and much higher for acid chlorides at 1800, because we don't have a large contribution of that resonance structure. So there's mostly double bond character. Okay, so NMR, the alpha hydrogens, just like before, were 2 to 2.5 ppm. the NHs of excuse me, NHs of amides were 7 to 8, and you can kind of think of this as because they are there is a major contribution resonance structure that has the N double bond C, so it's kind of shifted much more downfield. The CO is all the same, as pretty much the same as ketones and aldehydes, so we got 160 to 180 in the carbon NMR, and then CN is 115 to 120. Now, Shazam! We got interesting derivatives, and now I like this one because we got a lot of cool structures here. The top three, those are some esters that have some pretty big roles in making delicious foods. So the odor of banana is the one on the left. We got mango and pineapple. Those are esters, obviously. And vitamin C. Look at that structure. It's like a it's like a lactone, right? Because we got that cyclic ester. Cocaine got some esters in there as well. And another really cool one is antibiotics. So antibiotics have the beta lactam, remember lactamine, if you will, that structure within, and on the far left of this structure of this penicillin is an R group, and that's because there are subtle differences in the R group to give you different types of penicillin. There are also similar structures for amoxicillin and other antibiotics. Sorry, I lost that word for some reason. That structure right there, that beta lactam structure, is actually the reason why this group, or why antibiotics do their job. And we'll talk a little bit about this later when we get into the reactivity of amides. So this chapter is all about reactions. So brace yourself, because we're going to go over a ton and you're going to have to put in the work, because practice is what's going to make perfect. So the reactions of acid chlorides, first and foremost, is something we've seen before. We saw acid chlorides reacting with Grignards, and remember the Grignards were strong nucleophiles, and they attacked twice to make an alcohol, as well as the organolithiums. However, the organocuprates only reacted once, and so those didn't give the alcohol like the other two, but it gave the ketone. And that would be an acyl substitution product, analogous to what we're discussing in this chapter. However, the other two did actually do acyl substitution first, but they were strong nucleophiles, so they just kind of kept moving on. So, some new stuff now, though, right? So, acid chlorides, super reactive, and they will react with water. What's the outcome? Well, the nucleophilic attacks according to that general mechanism that we went over. And we've got this intermediate here, neutralization of that oxonium. And then the negatively charged oxygen can come down and kick out that chloride leaving group. And what that leaves us in the end is a carboxylic acid. So this was analogous to the nucleophile with the hydrogen on it, as we showed above, or a negatively charged nucleophile could be hydroxide, and that would give you the same outcome. However, if hydroxide were to attack, we would still get the formation of a carboxylic acid, but since hydroxide's present, we would remove the acidic proton of that carboxylic acid, and that would give us a carboxylate water as well as our chloride. but pretty much the same thing. So let's say we had an acid chloride as well as a carboxylate. What happens now is this is equivalent to a negatively charged nucleophile, so it attacks that oxygen, kicks back down the pi electrons, kicking off the chloride, and what we end up with is an anhydride. So this is one method to make an anhydride. Notice the chloride is there, and the pKa of its conjugate acid is -7. Acetate is acetic acids is the conjugate acid, and that pKa is 4. So the stronger acid means a weaker base and a better leaving group. So that's why this reaction can proceed forward, because the chloride is a better leaving group than, let's say, the acetate. So we have a couple other reactions that we can go over. You might be able to just immediately predict what's going to happen. We're just substituting the chlorine for for basically whatever the nucleophile is over the reaction arrow, and in this case it's an amine, and that's exactly what we get. So since the end product is an amine as well as an ammonium, because we needed something to remove that extra hydrogen on the tetrahedral intermediate, the second step, we needed 2 equivalents of the ammonia. And so whenever we're reacting an acid chloride with an amine, we need 2 equivalents to do this every single time. So let's practice. How do we do this? Or can this transformation actually proceed forward, would be the question. So pause it, think about it. This is going to be related to acid-base chemistry. Let's take a look at the pKa's of the leaving group's conjugate acid. Chloride -7, hydroxide 16, amide +38, and the alkoxide 16. So remember, we always want to move forward to form the weaker acid and base. And so that means we want to proceed from a pKa on the left of -7, or I'm sorry, very low, to a pKa on the right that's very high. So the bottom reaction actually won't work, and the top reaction does proceed. And this makes sense. Chloride is a good leaving group, and amide is a terrible leaving group. And chloride's a weak base, amide's a very strong base. So the reaction only proceeds to form the weaker acid and the weaker base. And I put it only in parentheses because, I mean, there's always exceptions to the rule, and that's specific to this amide reaction. We can get it to move forward as long as we do some tips and tricks. So let's go over the reaction of anhydrides. So anhydride, here you go. If we have a nucleophile, it's going to attack the most electrophilic carbon. Well, there's 2 carbonyl carbons here, and they're both nestled in between 2 oxygens, so they're very electrophilic. The nucleophile can attack, and you end up with pretty much a carboxylic acid or carboxylate, which I haven't drawn, and then we end up with a new carbonyl compound with a nucleophile on there. The leaving group in this case would be an acetate, but depending on if it's basic medium or acidic medium, we could get acetic acid or acetate. So the leaving group I circled in red, and on the right side I wrote it in red. Let's say we reacted our anhydride with water. I drew it in red so that we can kind of follow the protons. We would end up with 1 equivalent of a carboxylic acid that kind of took that water, and then the other one would be a carboxylic acid that was the leaving group. If we did the reaction with an alcohol, we can again make an ester as well as a carboxylic acid. So as you can see, the trend here for anhydrides is we have a nucleophile come in and then we lose a carboxylic acid as our leaving group, and then our new acyl substitution occurred as well. So we'll get whatever the whatever the rest of that is with the new nucleophile on that carbonyl carbon. Pretty straightforward reaction. This last one right here, remember amines are basic, and so this is where we might get an obvious formation of a carboxylate just by an extra equivalent of the amine coming and removing that acidic proton, forming an ammonium. All right, so some practical applications. Well, not for you guys because don't do this, but the structure I'm drawing is actually morphine. So that's why I'm saying don't mess around with this because we're about to make heroin on paper. And since it is an annoying structure to draw, I will just copy and paste. Sorry, let me move this to give us a little bit more room. So if we reacted morphine with acetic anhydride, we have to look for the most nucleophilic site on that, and we have two OHs sticking off that can actually attack, just like we saw above. The reaction of alcohols or even water would be an equivalent. Those two reacting with an anhydride pretty much results in the formation of a new ester or carboxylic acid. And so that's exactly what happens in the presence of acetic anhydride. And this turns out to be the structure of heroin. And this is also known as acetylation because we're adding an acetyl group or an acyl group. It could be called acetylation or acylation. And so that's just two different ways to discuss this type of reaction. All right, so let's put this knowledge to work now and do a couple of practice problems. So let's say we started with benzene. I'm not going to ask you guys to try and figure this out yet, but a couple steps will get us to this structure here where we have an alcohol as well as a carboxylic acid. Now, the question would be, which of the OHs is the most nucleophilic site? It would be the top one, and that's because the OH on the carboxylic acid is resonance stabilized. So that's actually where you would add the acyl group. So this is, again, an acetylation or an acylation reaction. Let's say we had this alcohol and amine functionality on the benzene ring. Which one's more basic or nucleophilic? That would be the nitrogen. It's further to the left on the periodic table than relative to oxygen. And so that's where the acyl group ends up. Okay. This is actually the drug Tylenol or acetaminophen, while the drug from above is aspirin. So the next set of reactions that we're going to go over are those of carboxylic acids. So let's say we had a carboxylic acid and we reacted it with a nucleophile like hydroxide or ammonia. Well, those are good bases as well. So what's going to happen is they're going to remove the acidic proton in the carboxylic acid to form water or ammonium. What about chloride? Well, hail to the no, because remember, chloride is an excellent leaving group. And when it comes to chlorides being good nucleophiles in SN1 and SN2 reactions, that worked out great because it was kind of a different story. When it comes to acyl substitution, chlorides are terrible nucleophiles, and that's because they are going to be such good leaving groups. And the intermediates are also, if we take a look at the acid-base chemistry associated with it, we can see that alkoxide in the tetrahedral intermediate is a stronger base. And then when it comes back down to kick it out, the chloride is a better leaving group, and so essentially end up with the carboxylic acid again, if it were even going to attack. And then in addition to that, the hydroxide is a stronger base, and so it's a worse leaving group. The reaction just falls apart. And by that I mean it doesn't proceed because nothing is working out to actually promote the reaction to move forward. So how can we form an acid chloride? Well, recall from earlier chapters, maybe chapter 9 or something, alcohols. Alcohols react with dianyl chloride to form alkyl halides. And we can do this in a similar manner with carboxylic acids and thionyl chloride. Recall the structure of thionyl chloride drawn above the carboxylic acid. That sulfur is electrophilic and can be attacked by the carbonyl oxygen, resulting in this tetrahedral intermediate. I like this mechanism a lot, by the way. It's super fun. And we kick that chloride off of the sulfur by creating the sulfur oxygen double bond again. We have this oxonium intermediate. Since there is a carbon-oxygen double bond and that oxygen is positive, that carbon is susceptible to nucleophilic attack, right? Sorry, I'm highlighting all these chlorines in red now just so we can keep track of where they're going. That chlorine now attacks that carbonyl carbon and neutralizes that oxonium, and we have another tetrahedral intermediate. And so what do we need to do in order for the reaction to proceed as we want it? Well, we need obviously the sulfur group to leave, but we also need one of those oxygens to leave as well. So let's force something to leave. And the way we do that is create that CO double bond. And that CO single bond, those two electrons actually go to create an oxygen sulfur double bond, which ultimately kicks out the chloride. This forms sulfur dioxide chloride ion, as well as pretty much our acid chloride. But we do need to remove that last hydrogen. So the reason why this reaction actually proceeds so nicely is because sulfur dioxide is a gas, and since it's a gas, this reaction is actually entropically favored. And usually gases are amazing leaving groups. So that's exactly what happens. This chloride can come along and remove that that acidic proton on the oxonium, and that ultimately gives us our acid chloride product as well as HCl. So this is also a pretty gnarly reaction, and one, because you make sulfur dioxide gas, two, because you make HCl, and then three, because you make an acid chloride, which is very reactive with water, and that is water in the atmosphere, and it makes more HCl gas after the fact, after it reacts too. So it's pretty fun. All right, let's take a look at somewhat of a multi-step sequence. So what we have here is 1, thionyl chloride, 2, an amine in excess. This is diethylamine. So we have to know that the first step for the addition of thionyl chloride with carboxylic acid makes an acid chloride, right? And then after that, an amine just substitutes the chloride for, and then ultimately making an amide. And so these types of reactions should be pretty easy for us, but we just have to be aware of what each reagent does. Otherwise, we're kind of screwed in the end. So get to know those reactions really well so they come to you very quickly. So the formation of anhydrides. So we saw how to make anhydrides before. We can make them another way though. So if we have a dicarboxylic acid and we heat the crap out of it, well, what we end up getting is the loss of water and the formation of an anhydride. That's why it's called an anhydride, because it's dehydrated and it lost all water. The acid chloride route is also another one that we can do or use to make anhydrides. We already went over the reactions of those anhydrides, so we're pretty much done with those guys. Fischer Esterification. So this one is super fun as well because it makes some really nice things. So the interesting compounds that I went over earlier were esters, and they smelled good. Banana, mango, they were in our food and stuff like that. Banana Laffy Taffy. And so we have, the way it works is we have a carboxylic acid, an alcohol, and an acid. So if we have an acid, we want to protonate something, right? Which is the stronger base, the carbonyl oxygen or the OH on the carboxyl group. That's going to be the carbonyl oxygen because it is resonance stabilized. So it makes a weaker conjugate acid, and that means it is a stronger base. So let's do that. Let's protonate. Once we protonate, we end up with an oxonium intermediate that makes the carbon very susceptible to nucleophilic attack by what? The nucleophile, the alcohol, that attacks. We get a tetrahedral intermediate with an oxonium on that alcohol that we added. We can do a proton transfer, get rid of that on the that hydrogen on the alcohol, and add it to one of the other OHs, because now we have a good water leaving group, and that's what we do. We form that CO double bond to kick the water out, giving us a oxonium intermediate again, one step away, just a deprotonation step away from our final product, the ester. So this is ethyl acetate. Ethyl acetate, if you noticed or didn't notice, is it came from ethanol and acetic acid, right? Ethyl acetate's actually in nail polish remover, it's in glue, and it's also in wine. As I said, it's acetic acid and ethanol make it. Well, acetic acid is also known as vinegar. So it's also the oxidation product of ethanol, and that's why it forms in wine, because the presence of oxygen breaks it down or builds it up. Depends on what you're trying to get. So we also have the ethanol coming in, and the source of acid is going to be the organic acids from the wine, or those that came from the grapes. So the reason why I like the Fischer Esterification is because it makes a lot of really good esters. Esters are really nice smelling compounds. If you think about it, acetic acid, doesn't smell very good, right? Vinegar is not an extremely pleasant smell, but when you combine it with this alcohol, ethyl acetate has a nice smell. it's actually kind of sweet. You know, it's a nail polish remover and glue, but I don't know. Some people like sniffing that. But if you think about these molecules right here, this top one, if you combine that alcohol with essentially, I'll just call it vinegar, right? You end up getting a banana smell, which is crazy to me because you have this not so pleasant smelling liquid, and then you jam the two molecules together without not a totally different structural uh, change, but a very different smell. And that's why I like this Fischer Esterification reaction, because it shit's bananas, you know what I mean? Bananas. You also have apples if you want. Um, Pentanol with vinegar. Um, Butanol, or whatever, how many is in that oranges one. You can get pears and oranges. So you should be able to go forward and backwards. So what alcohol and carboxylic acid were needed in order to make the pear and orange smelly ester? Well, that's pretty straightforward. I mean, pretty much the red unit, that's the source of the alcohol. Just add a hydrogen on there, and then the black unit on that structure is the carboxylic acid. All right, so another cool example of hydrolysis of an ester, as well as esterification or an acylation, is converting oil of wintergreen, so the wintergreen gum and flavoring that you have in your mints, and converting that to aspirin. You can see the ester on the top part was converted to a carboxylic acid, and then the bottom portion underwent an acylation. So this was actually a general chemistry experiment that I remember doing. I don't remember why I was in Gen Chem, but I remember loved doing it because I liked wintergreen, and then we made aspirin, and I thought that was awesome. Maybe that's what got me lured in a little bit more to chemistry. So another really cool esterification is the intramolecular stuff. So here's another example of intramolecular esterification to form a lactone. And I've highlighted the different oxygens in red and blue just to indicate where they end up in the final product, just to help us out. Because when we have a carboxylic acid normally, in the presence of an alcohol, the OH from the carboxylic acid typically leaves, and then we have the alcohol attacking the carbonyl. And that's exactly what's going to happen here when in the presence of a strong acid, or a little bit of an acid. So H2SO4 protonates that carbonyl oxygen, and then making that carbon more susceptible to nucleophilic attack. We now have what looks like the ring structure of our final product, but we do have a couple extra oxygen, or a couple extra things on that carbon. So what we want to do is do a proton transfer to get us to where we need, or to where we have a good leaving group, water, create that carbon-oxygen double bond, and then deprotonate that oxonium in the end. And that's pretty much it. I like to keep track of my carbons by numbering them. That way it makes for an easier synthesis or formation of that mechanism, and I would suggest you do that as well. So we've mentioned before that this is all in equilibrium. We just need a catalytic amount of acid, because notice that the H plus that was added in the beginning actually forms in the end after that final deprotonation of the oxonium. But since this is all in equilibrium, we can't actually go backwards. We just use Le Chatelier's to drive that forward or backward. So hydrolysis of esters can actually occur in the presence of acid, which is what we've seen, what we can see above. That would be the reverse reaction of the mechanism above. But it can also be done in the presence of base. So a hydroxide can come in and attack that carbonyl oxygen, and the mechanism looks exactly as normal, but that kicks down. It's basically just a two-step process, right? Then in the end, we actually deprotonate that carboxylic acid to form the ethanol in this case, as well as the carboxylate. Now, there's another term for this, and that's called saponification. This is also something that I mentioned before in lecture. So if you heard me talking about Fight Club, that's exactly what I was talking about. So they take fats from they take fats from like a liposuction place, and then they basically make soap in their bathtub using lye, which is just sodium hydroxide. And so this is basically a triglyceride or triacyl glycerol, and it's basically glycerol combined with 3 fatty acids, and those are all esters, and so they undergo hydrolysis in the presence of base, and you get 3 carboxylates. And remember, these 3 carboxylates actually form the micelle for soap. So there you have it. Now you can make soap in your bathtub. No, don't do that. So, formation of amides. Amides can be made in a number of different ways. Since we're on the topic of esters, or the ethoxide leaving group in this case is actually a better leaving group than the amide, and so the nitrogen nucleophile can actually come in and attack and force that out. Just like any other nucleophile that would have come in. Carboxylic acids in the presence of an amine, however, that just undergoes a acid-base reaction, and you get a carboxylate as well as an ammonium. But in the presence of heat or like other harsher conditions, you can force that reaction forward to get the amide as well as water forming. Or to get more reasonable temperatures or reaction conditions, you can convert a carboxylic acid to an amide using something called DCC. So DCC is written above the reaction arrow and is dicyclohexocarbodiimide. So that's actually not the structure on the right. Sorry, that looks a little confusing. But the structure on the right is a urea as a result of the reaction. It's a reaction product. And so the role of DCC is to make OH a better leaving group. That's the role of a lot of things, right? When we're trying to do these reactions, transformations. And so this is DCC. We have the 2 dicyclohexyl groups. That nitrogen has a lone pair and is basic, so that removes the acidic proton on carboxylic acid, forming a carboxylate as well as an aminium type molecule. That carbon drawn in blue is delta positive. It's very electrophilic because it's nestled in between those 2 nitrogens, and it's susceptible to nucleophilic attack by the carboxylate, which is what happens, and that neutralizes the nitrogen, and resulting in this intermediate here. Now we have something that's no longer an OH group, so we have this oxygen with this large group hanging off of it that is a much better leaving group. So that carbon is going to get attacked by the ammonia, resulting in this new tetrahedral intermediate with a positive charge on the nitrogen and a negative on the oxygen. A proton transfer will occur, removing the proton on the ammonia that attacked and putting it on the iminium, or the nitrogen doubly bound to the carbon. Now that oxygen forms that carbonyl group of the amide, that breaks that carbon-oxygen single bond, creating another carbon-oxygen double bond and neutralizing the nitrogen on the product of the diamide. Now this new product is called dicyclohexylurea, and we also have our amide. So the product on the right, even though it looks kind of fancy, is useless. The one on the left is the desired amide product. But the presence of DCC allows us to do these amide transformations at much lower temperatures and just easier conditions that are more friendly to other functional groups in our molecule. All right, so we have acetaminophen on the left and aspirin on the right. Acetaminophen is shelf stable while aspirin eventually decomposes. Now, think about why that would be. Well, we have an amide functional group, and then on the other aspirin, we have an ester functional group. Well, we just talked about how strong those amide bonds are and how we need to get harsh conditions like 100 degrees or higher. But the esters, those can easily be broken down in the presence of just a little bit of water. And so that's why over time the aspirin decomposes while the acetaminophen is stable. All right, so let's take another look at a lactam in the presence of hydroxide in water. What's going to happen? What is the final product going to be? Remember, we need a lot of heat to break that amide bond. So typically, these lactams, they are more susceptible to decomposition because they are in a ring structure. This one is an alpha, beta, gamma, delta lactam, and so it is a 6-membered ring, and it is going to be a little bit more stable than, let's say, an beta lactam, which is a 4-membered ring. Or, yeah. And so we know that the hydroxide is going to attack that carbonyl carbon, and once that oxygen brings those lone pairs back down, we kick off nitrogen having a negative charge as a leaving group, and then ultimately what we can do is get a proton transfer, protonating that amine or protonating that amide to an amine, and forming the carboxylate. I like to keep track again of my carbons to help make sure that the final product is drawn correctly. So, as I mentioned before, a beta lactam is actually more strained because it's a 4-membered ring. So, if I were to ask you, out of this particular antibiotic, we have 2 amide functional groups. Which one would be more reactive? You should tell me the beta lactam because of that ring strain. And so this is actually the way antibiotics work. If we have an antibiotic in the presence of an enzyme, if we have that enzyme is actually making up the glycopeptides or the cell walls of bacteria, then it reacts with amides. And if we have this beta lactam present in the antibiotic and it reacts with the enzyme and binds pretty strongly, then that enzyme can no longer do its job of creating the cell wall of the bacteria. And so ultimately we've inactivated this enzyme and we are destroying the bacteria. And that's pretty much how antibiotics work. That's very simply put, but I mean, that's the chemistry of it. The reactive strained ring in the antibiotic, the beta lactam, is the source of that reactivity and is the reason why we can shut that down. So, all right. So, natural and synthetic fibers. So these are pretty cool. We are very familiar with polymers already. Wool and silk, as well as cotton and linen, those are both naturally occurring polymers. Wool and silk are they're called polyamide. And then cotton and linen, those are actually just repeating units of glucose. So that's just cellulose, right? So depending on the three-dimensional structure, you can get cellulose, amylopectin, or amylose, and those are the starches as well. So pretty cool stuff. Now, the synthetic fibers that we might be familiar with would be like nylon. So nylon 6,6 is also a polyamide and consists of this unit here. This is specific to nylon 6,6. Highlighted in blue, I have the amine portion, and then highlighted in red, I have the carbonyl source as well. And so if we wanted to figure out how to make this, well, one of the easiest ways to make amides is with an amine, and so that's why I've drawn that in blue. And our carbonyl source would be an acid chloride, because that is in fact the most reactive carbonyl that we have access to. And so that's exactly what happens. You have one unit of the amine reacting with one side of the acid chloride, and then we have another unit coming to attack the other side. And as you can imagine, this can keep building into a long chain, and that's why it is called a polymer. And we have the longer it gets, the longer the strands. It's called nylon 6,6 also because each of those monomer units, the amine as well as the acid chloride, they have 6 carbons in them. You can get nylons with longer chains in between. So polyethylene terephthalate, or PET, that is a polyester that we see in everything, right? And it looks fancy. It looks like this. So since it's a polyester, what we want to think about is what an ester is made of. An ester typically starts from a carboxylic acid, and in the presence of acid as well as an alcohol, we can form an ester. So if we look closely, we have our carbonyl group right here. This is going to be the source of our carboxylic acid, as well as the red unit, which would be the source of our alcohol. So keep in mind, when we're doing these polymers, we have to have two sides that can react. That way we can have this chain building larger and larger. And so, as you saw in the amide and now in the ester, we have a dicarbonyl group. So in this case, a dicarboxylic acid. And then we had a diamine in the amide, and now we have a diol, double alcohol. So this will just take a little practice, but there are some homework assignments that'll help you with that. And another little quick ditty that I wanted to introduce to you is some more bio stuff. It's an acetyl coenzyme A, or aka acetyl-CoA. Acetyl-CoA is a thioester, and that SCOA, that coenzyme A, is a good leaving group. And so in the presence of a nucleophile, we can attack the carbonyl and essentially kick out that SCOA and create this new compound. It's an ester, and it is acetylcholine. Then this is a neurotransmitter. And so as we go over these biological applications, I want you to keep in mind what classes you'll be taking in the future. So I have a previous student that just took 2 quarters of biochem at UCSB, and he was telling me how understanding OChem is essential. It's also actually, it's not necessarily essential, but it's going to help you be more successful in biochem. He just shot me over a quick little text of the information that he recalls being super helpful, and that was understanding front and backside attacks, the racemic mixtures, and how stereochemistry is important for enzymes, isotopic labeling of carbons, lots of carbonyl chemistry. So, I mean, we're showing you right now the carbonyl chemistry, right? Specifically ketones and aldehydes, he said, but you'll see it in everything, as I just showed you with this neurotransmitter acetylcholine. Obviously, arrow pushing and redox reactions are going to be important as well. So knowing the nucleophile is attacking the electrophile and identifying those spots in there. He did also tell me a little bit about how, because I wanted him to touch up on cell biology and how important that was. Obviously, it's going to be a huge part in it, but I wanted more specific information on how much of the information you went over in cell bio is applied to UCSB's biochem. I mean, I was kind of a silly question, but he said, yeah, a ton of it. And it almost seems easier at UCSB. So that's good. We're preparing you well here. And it's also going to be really useful for genetics, using the ideas of recombinant DNA and E. coli for reuptake and stuff like that. So just wanted to let you know. Oh, he said biochem uses all the amino acids, obviously. So we just went over those, and I would encourage you to kind of better understand those. And I mean, if you wanted to, even memorize them, because I know that might be a thing eventually. All right, so back to chemistry. The next practical application would be cholesterol esters. So it's the same thing with this S-CoA, this coenzyme, but we have a longer chain on that carbonyl group. And what I'm going to do is draw out a cholesterol or a steroid here. So just bear with me 1 second. There's a lot of stuff going on. So one thing, if you haven't noticed, the steroids are typically going to have that general ring structure with a couple things popping off on either side. So if you see those, you're going to think steroids. So this steroid has a hydroxyl group on there that can attack the carbonyl group, just like we saw before. And what you end up with is this larger structure on the bottom, and this is called a cholesterol ester, because the steroids are the cholesterol, and we have an ester now, right? So this is actually the cause of plaque buildup in LDLs, or low density lipoproteins. And so yeah, you could probably check out your book. There's a cool image in there, and a little small explanation of how that might work. But you have your LDLs and your HDLs. LDLs cause they're the bad cholesterol that cause the plaque buildup, and the HDLs kind of swoop on that and bring it back to your liver to be processed to create more steroids. All right, so let's get some practice in. So we have an acid chloride, and we have C6H5CO2Na. So what we need to know is that reagent on the top of the reaction arrow is a carboxylate ion with a sodium countercation. That is the nucleophile that can attack the carbonyl, the acid chloride. We have this intermediate, which then comes down and kicks off that chloride leaving group. And what is our functional group in the end? What do we have? We have an anhydride. Remember that the benzene ring can be abbreviated as a pH when you have it as a substituent. All right, so let's say we had a carboxylic acid. And we have, what I've done here is I'm drawing 4 different reaction arrows because we can create many different things from one carboxylic acid. So in the presence of acid and ethanol, we are going to be doing a Fischer Esterification. This should pop in your head very quickly. You should be able to quickly jump to the product. When you have it in the presence of thionyl chloride, this is, boom, think acid chloride, just jump straight to that. Sodium hydroxide, this is just an acid-base reaction. So you deprotonate the carboxylic acid and make a carboxylate. Lithium aluminum hydride, we saw this a couple chapters ago, and this is just a reduction. 4 days. Remember, lithium aluminum hydride just reduces the crap out of everything, including carboxylic acids to an alcohol. So this type of stuff, although we should know the mechanism, should also just come very quickly to us. So this particular ester in the presence of a strong acid followed by, in a next step, CH3COCl and pyridine. We have to know that that is an acid chloride. Okay, so, and pyridine is going to be a base. And this base is to neutralize the intermediates in the mechanism, which we'll see. So, or which we saw before. So H3O plus in the presence of an acid or ester undergoes hydrolysis to form methanol and a carboxylic acid. Pyridine can remove the acidic proton of the carboxylic acid. That carboxylic acid becomes a carboxylate, which can now attack very easily the acid chloride, and what we end up with is pretty much a multi-step sequence to get to an anhydride. All right, so let's take a look at another problem. What we have here is a cyclic ester, also known as a lactone. In the presence of us H3O plus, we should know that hydrolysis is going to occur. And when hydrolysis occurs of an ester, we have a carboxylic acid forming as well as an alcohol. In this case, it's within the same molecule. And so this is this type of stuff is going to take some practice to get directly to the product. But it is pretty straightforward once you get the hang of it. Now let's take a look at another problem and see how we might be able to get from this bromotoluene on the left to the molecule with the amide functional group on the right. This is DEET, so the bug spray. We know that in order to make an amide, we have to go backwards and react. The easiest way is an acid chloride and an amine. So in this particular case, we would typically be able to just add the amine. We're not asked to make that. So, but we do need to know how to make that acid chloride. We know we make that from a carboxylic acid, and we know that we can go back from a bromobenzene and make a carboxylic acid with a Grignard. The way we do that is add magnesium. That makes the Grignard. And that Grignard attacks carbon dioxide, and then followed by acidic water, we can make a carboxylic acid. Carboxylic acid with thionyl chloride makes the acid chloride, and then throw in the amine, 2 equivalents, and we get our DEET final product. And then also that second equivalent of the amine was to neutralize, and we get an ammonium from that. So that's just another example of a multi-step synthesis that we should be able to handle pretty easily, eventually at least. So this one is a more complicated mechanism, and it's still pretty straightforward, and we understand. I think the difficult part is probably keeping track of all the carbons So again, number those if it helps you. It should help you. And ester, what we see, the cyclic ester in the presence of a strong acid, we're just going to protonate that carbonyl oxygen, right? Just do what you should do first. We have an acid, we have an ester, protonate that carbonyl oxygen. Then what's going to happen? We make an oxonium intermediate that has a carbon that's susceptible to nucleophilic attack. So what are we going to attack with? We don't have anything else other than the alcohol that's on the same molecule. So attack it. Okay, and that, again, this is where it gets weird, keeping track of our carbons, but if you have to draw the structure a little off, that's okay. We have this oxonium intermediate now. A little proton transfer will take care of that oxygen charge, but transfer it to another oxygen, the one that we want to leave. Remember, this is very analogous to all of the other ester mechanisms. The original carbonyl always stays, the carbonyl oxygen always stays, while the other one leaves. So we protonated that, we made an oxonium, created that carbon-oxygen double bond, and kicked out the alcohol. Now I've drawn it in the same structure just to help us keep track of everything. And then now I labeled all of the carbons to figure out how many carbons are in that ring, so that I can draw like a normal cyclohexane type ring. And then we see that on carbon number 3, we have the R group coming off of it. So 1, 2, 3, boom, draw that coming off. Sorry, I remember that is a sigma bond, so it can rotate, but I redrew it just to make it match the original. All right, so this right here, I put together for you guys. I'm not sure if it's even helpful. It took me a long time, and I was like, damn, I'm not sure how helpful it's going to be, but it's there. So if you want to use that chart, you can. The last little thing that I came across was something that I found really interesting. My wife bought some flowers from Trader Joe's, and they're called Persian Buttercup. So I'm not actually sure if that's what the ones she got from Trader Joe's are, but I used an app to take a picture to figure out what flower it was, and that's what it told me. So, and a little fun fact about those is they're toxic. So that's cool. Let's just go ahead and buy those at the grocery store and then bring them home to my family. Well, ranunculin is the molecule within the flower that is broken down into the toxic substance. Pretty fun to look at this structure because it consists of a glucose molecule, pointing out an acetal, just for a reminder for you guys. And we got this cyclic ester popping off of it. Pretty neat. But if you imagine in the hydrogen there, if you were to remove that with a base, we would deprotonate that, and those electrons could go to the carbon bond, and that kicks off the glucose as a leaving group. So this is an enzymatic process. So that's not necessarily how it might go, but that's the general idea. And then we form protoaneminin, which is toxic. Uh-oh. You know what else is toxic? Britney Spears. Don't you know that she's toxic? Oh, snap. It's Britney, bitch. Or it's protoaneminin, bitch. So this thing right here, it still is a cyclic ester, right? And if you actually leave the flowers to dry out, that protoaneminin decomposes. So if you leaf out to dry, check that out. That's a little just off the top of my head. It's perfect. It actually dimerizes to form this crazy structure right here, which ultimately, again, if you leave it out to dry, it's just able to react with the oxygen in the air as well as water in the environment of the plant and perhaps other enzymes, and it actually breaks down into this carboxylic acid as well, which then after that plant is dried out, it is no longer toxic because protoanemone is no longer present because it broke down. Cool stuff, huh? All right. So that's pretty much it for chapter 20. I know there's a lot of reactions in there. Those are just going to take some time and practice, but I am here to answer any questions for you guys. So please let me know if you have any questions. The homework I'll be posting is right here. So take a look at that, record those. Oop. I'm sorry. The homework problems are here, so make sure you take care of those and you get your practice in, and then let me know if you have any questions. I can put together some videos to answer those. Don't forget, we do have an exam coming up as well. So good luck, have fun, do your thing.